Okay, I've gone over how to do this a couple of different ways. And I think what we're going to do is we're going to list the receptor types on one end, like I've started over here, and we're going to list the possible effectors on the other end of the board, and we're going to try and connect these two by the end of this lecture. And this is kind of the middle picture that connects the two things that I'm trying to draw. We've seen something like this. In fact, I pointed out the exam question where we were drawing parts of the spinal cord last time. We said sensory information always comes in through the dorsal root. There's that dorsal root ganglion coming into that posterior gray horn. We can see the green interneuron within the gray matter doing the information processing. And then we've got our motor neuron that has its cell body in that anterior gray horn. And then the axon portion leaves out of the ventral root and goes all the way out to the effector. In this case, that effector is skeletal muscle. So we'll be sure to list skeletal muscle as one of the effectors. Let's start over here on this end with the receptor end. So receptors are also out here in the peripheral nervous system. And we can list receptors based off of, we're going to list 3 different types of receptors. I just ripped open a blister, but that's all right. I'll use this brown color, and we're going to list different types of receptors. The first category is listing the type of receptor based off of its stimulus. So if we think of stimulus type, which is one way in which receptors are named, we're going to see photoreceptors, which pick up photons of light. There are mechanoreceptors. So photoreceptors are just light. Mechanoreceptors, this is going to be some type of physical force. I'll explain mechanoreceptors. We looked at a hair cell earlier, and depending on which direction the hairs on that hair cell are bending one direction or the other, they can either send excitatory signals or inhibitory signals. I'll say directional force. That might be more descriptive. So directional force. Another receptor type is going to be thermoreceptors. Hello? And as you can guess, these are receptors that pick up different temperatures. So you can tell if you're walking across this floor in your bare feet, whether the floor is cold or whether it's hot. Fourth category is going to be chemical receptors, these chemoreceptors. Chemoreceptors are all over the body. They're in the bloodstream that can tell whether your ratio of carbon dioxide is too high or too low. And it can trigger whether or not you start to hyperventilate or hypoventilate. If you're blowing off too much CO2 and you need to hold on to it, your lungs will involuntarily cause you to slow down your breathing so that you don't blow off that excess CO2 and you hold on to it. So we'll say monitor chemical levels. I can spell that. Monitor chemical levels. I'll do things like, give examples like oxygen or glucose or calcium ions. We could go on. pH. All examples of these chemoreceptors. And then the last one we're going to list are nociceptors. And these are sensitive to pain. Pain receptors. Ooh. I've got just enough room. I'm gonna do something with the board. I'm gonna try and separate it into 2 remaining sections. And in one of these sections, I've got just enough room to list another category of, it's the same receptors, but it's basically categorizing them based off of their location. So we'll say location. If we want to classify receptors based off of their location, there are some receptors that are around the surface of the body. These superficial types of receptors are called exteroceptors. These, again, are mostly going to skeletal muscles. We talked about if you step on a tack or slam your hand in a car door. These are the types of superficial sensory receptors, Meissner corpuscles, Pacinian corpuscles, free nerve endings. These are all in the surface of the body versus these interoreceptors, which are inside visceral organs. So if you have a stomach ache or one of those things, heartburn or any of those types of sensations that are coming from an internal organ, these are picked up by interoceptors. Then the last one that we're going to list based off of their location, there are receptors called proprioceptors, and they're located in the joints of the body. So you can tell, for example, if your arm is behind your back or my knee is twisted into a weird position, you've got these sensory receptors in the joints of the body that are relaying information about the position of those joints. These are proprioceptors. We'll say external, internal, and then joints of the body. Then the last category, our last way in which we're going to group these types of receptors are based on their complexity. So for our third option here, we're going to list them based off of their structural complexity. These only have two categories. We're going to call these either complex, which is going to be the case for special sensory. We're going to draw some of these complex receptors that pick up things like photons. We're going to draw the photoreceptors in the eye. We're going to pick up the sound wave receptors in the ear. Then we're going to list what are called just simple receptors. These simple receptors are going to be located in the exterior part of the body, and we can break them down based off of whether they are encapsulated, kind of wrapped in connective tissue. I'm thinking of those Meissner corpuscles without going and grabbing that. Meissner corpuscles and Pacinian corpuscles are what we would consider as encapsulated. Again, wrapped in connective tissue, or they are unencapsulated. An example of one of the unencapsulated nerve endings would be like a free nerve ending, or one of those nerve endings that wrapped around the base of the root of a hair. There was a root hair nerve plexus, and those were unencapsulated nerve endings that just wrapped around the root of a hair.
Encapsulated was wrapped in myelin sheath?
Yes, there's some type of connective tissue that wraps around that receptor, like a Meissner corpuscle or the Pacinian corpuscle that had those multiple layers of connective tissue. It kind of looked like a fingerprint. Okay, I'm going to come over here while we're on this, and I'm just going to list some possible effectors. And let's see if I can do this in a way that allows us to keep it on the board. I'll use these two colors. One of these effectors, let's make this light pink color. I'm going to make this a skeletal muscle. And how we know it's a skeletal muscle, I'm going to draw this thing that's got a depression and then another smooth surface. This was our skeletal muscle fiber. Skeletal muscle fiber is the same thing as a muscle cell, skeletal muscle cell. So we've drawn one of these skeletal muscle cells. It has this area of interest that we said was the motor end plate. And on that motor end plate, let's just get a color for it. Kind of like the dendrites and the cell body, there were these little receptors for neurotransmitters, those non-specific ion channels. And just like the ones on the dendrites in the cell body, they were binding acetylcholine. And if acetylcholine was present and binding to those receptors, it caused the channel to open up. If enough sodium ion rushed in, then it made it all the way up to the sarcolemma, where we had the voltage gates. I'll just put that visual up there real quick. There it is. As soon as we got up to this smooth sarcolemma is where we had our voltage gates for sodium and voltage gates for potassium. So down here, this motor end plate, we said this end plate potential is a type of graded potential. It's dependent on chemical signals, and if there is enough chemical signal. If there's insufficient chemical signal, then the little ripple never gets out of this depression. If there's sufficient chemical signal, then it actually triggers that first voltage gate, that first voltage domino to fall over. And once we involve these voltage gates, this whole part of it is where we begin the action potential. Anyway, we're going to see how these skeletal muscles are controlled as one of the effectors. For the other one, I'm just going to put a simple box that shows, it's going to show several things. This box down here, I'm just going to list inside. This box is going to represent things like cardiac muscle. So we're going to list things that are effectors other than skeletal muscle. So cardiac muscle, we'll say smooth muscle. I'll add glands. That can be digestive glands as well as sweat glands. And then I'm going to list adipose tissue. Adipose tissue, when we were talking about tissue types, we said this is where you store long-term energy. So if you, after eating a big meal, if you have excess nutrients, your body will store that excess nutrients as adipose tissue. And if you are at a time where there's not enough nutrients, your body can actually take that adipose tissue and build sugars out of it. It can break down that adipose tissue and use parts of it to construct its own sugars. It's something that's called gluconeogenesis, building new glucose molecules out of things like proteins or lipids. It's not without its problems. If you're building glucose molecules out of adipose tissue, this thing is going to be kicking off a bunch of hydrogen ions into the bloodstream.
So everything in that box you said was an effector?
Is an effector. Yeah, I'm trying to put this under the list of effectors. We've got skeletal muscles, then we've got cardiac muscle, smooth muscle, and glands. Just as a review and a preview of something that we're going to talk about, we said the things like cardiac muscle, smooth muscle, and glands, they were controlled by the autopilot, that autonomic nervous system. Because we're going to go there too at the end of this lecture. And if we're thinking of the autonomic nervous system, it has everything to do with these effectors. Right? The autopilot controls cardiac muscle, smooth muscle, and glands. You can't just think, all right, I want to make my sweat glands turn on or turn off. You can't do that. Or I want to make my heart rate increase or decrease. The autopilot does that. So we're going to see what are the effectors that control cardiac rate and cardiac muscle, smooth muscle, or whether or not we are sweaty or our digestive glands are activated, or whether or not we're adding adipose tissue or breaking down adipose tissue. We'll do this. What's interesting, the same, I'm getting to the hard part of the lecture at the very end. We're doing it right now. We're going to plant this little seed. And if I can keep this box on here the whole time, what's crazy is that you have what's called dual innervation. The fact that these same effectors, this same box of cardiac muscle, smooth muscle, and glands, it can be controlled by the sympathetic division. Does anybody remember what the sympathetic division would do to the cardiac muscle, smooth muscle, and glands? Sympathetic was fight or flight, right? And so the sympathetic division would cause this entire box. Cardiac muscle would increase. Your heart rate would increase. Smooth muscle, that directs blood flow. So if you were in fight or flight, smooth muscle is going to constrict around the blood vessels that go to the digestive system. It's no time to digest food if you're fighting or trying to get away from something. And at the same time, some of the smooth muscle is going to dilate on those blood vessels that go to the heart wall, because the heart, as it beats harder and more forcefully, it's going to need fresh blood flow. So these blood vessels dilate that go to the wall of the heart. Also, blood vessels that go to skeletal muscles dilate because you might need to run away or engage a threat. We don't have enough blood to be in all of the organ systems at once. So if we're in fight or flight, blood flow is directed to skeletal muscles and to the heart and to the brain and away from things like the digestive system. Glands, on the other hand, if we're in fight or flight, you're going to see your palms are going to sweat so that you can grip onto things. At the same time, digestive glands are going to shut off. It's no time to digest food. And then adipose tissue, if you were in fight or flight, adipose tissue is going to be melted away. Your body is in, thinking things very quickly. You're taking in information. This increased processing power requires more nutrients, so adipose tissue is liquidated. All of these things are going in one direction with sympathetic. How these are controlled by the sympathetic nervous system, there are receptors on here that I'm going to list them in a red color, and I'm going to call this entire category of receptors adrenergic receptors. So adrenergic receptors are receptors that bind adrenaline. So adrenaline molecules are things that are going to be increasing heart rate, fight or flight. So adrenergic receptors, I'm going to draw a few of these adrenergic receptors. We're going to see they have, again, I'm going to list them here. We're going to see there are 5 of them that we need to know. Alpha 1, let me try this again. I'll say alpha 2, then we'll say beta 1, beta 2, and beta 3. We've got these little, I can maybe draw them as little Y-shaped receptors. And these receptors are located on cardiac muscle, smooth muscle, and glands. And these are the types of receptors that are able to bind adrenaline molecules. Adrenergic receptors, the adrenaline molecules that we talk about are going to be either what's called norepinephrine or EPI for epinephrine. I believe it's on the back side of your lecture handout. You've got all of those. Maybe the second page of that, where you can see the different types of chemical signals that are going to be sent by, this is getting us into the autonomic nervous system discussion, because the autopilot, that autonomic nervous system, controls cardiac muscle, smooth muscle, and glands. I was just about to say, what's really interesting is that the nervous system can control these same effectors in 2 different directions. If we are in fight or flight, so adrenergic receptors, I'm just going to put a little note here. This is what would be triggered if we were under sympathetic stimulus. Sympathetic stimulus is going to involve these adrenergic receptors. The chemical signal is epinephrine or norepinephrine, and it's going to dock to these adrenergic receptors. We're going to talk about the different types of adrenergic receptors and where they are located. But what's interesting is at the same time, these same glands, if I switch colors here, I'll switch to this color. This is a darker blue color. We're going to see that there are receptors also on the same effectors that instead of binding epinephrine or norepinephrine, they're going to bind acetylcholine. And I feel like I might run out of room. I can't push that too far. Let me see if I can squeeze some information. I'm trying to draw these receptors. These receptors over here are going to bind acetylcholine. So that's their chemical that binds these receptors. So since they bind acetylcholine, a name for this class of receptors are called cholinergic receptors. So cholinergic receptors are going to be located in two places. Cholinergic receptors always bind acetylcholine. That's where they get their name. We're going to see some acetylcholine binding to these receptors on cardiac muscle, smooth muscle, and glands. These types of receptors, I'm going to make that a little bit smaller. We know that's acetylcholine. These types of receptors that we see are going to be called muscarinic receptors. Muscarinic receptors. This one, same thing, muscarinic. These muscarinic receptors are located again on cardiac muscle, smooth muscle, and glands, and they bind acetylcholine. You'll notice another cholinergic receptor. So if we're thinking of cholinergic receptors, just as a group, we're going to see there are two types of cholinergic receptors. These muscarinic receptors, located on cardiac muscle, smooth muscle, and glands. The other cholinergic receptors are located right here on the motor end plate. These, I'm going to throw them in here, these are called nicotinic receptors. That's really all of the detail for the receptors that we have to label. I thought maybe listing it early might be helpful. Let's connect the dots here. So what we're doing is we've listed the types of receptors, things that can pick up some type of stimulus and generate a signal by the sensory neuron. We know how sensory neurons talk to interneurons and interneurons talk to motor neurons. We'll now see how these motor neurons communicate to some of these effectors. Again, the top branch, we have voluntary control of skeletal muscles, and then the bottom pathway here, we have involuntary control of the cardiac muscle, smooth muscle, and glands. If we're under that sympathetic stimulus, it's adrenergic receptors. You know, the heart rate's getting ramped up. Smooth muscle is sending blood to the heart and skeletal muscles. Glands, like sweat glands, are activated. Digestive glands are decreased. We're burning through adipose tissue. But if we switch gears over here, and I know I can't back this thing up any further, but if we're instead of sympathetic stimulus, if we wanted to involve this parasympathetic stimulus, if we had some type of parasympathetic stimulus, then that is going to be sending acetylcholine. And that acetylcholine is going to be docking to these receptors, also located on the exact same effectors, cardiac muscle, smooth muscle, and glands. This is how you can get that other part of the autopilot, this, what did we call the parasympathetic division? If sympathetic was fight or flight, what was the parasympathetic division? It was rest and digest, that's right. And that's interesting how this opposing signal can cause the same effectors to do something different. So after you eat a big meal, if you're laying on the couch, you're getting hit by this rest and digest. So the cardiac muscle, that same cardiac muscle that was ramped up during adrenergic stimulus, during cholinergic stimulus, that heart rate is going to slow down. Smooth muscle, again, after you've eaten a big meal, that smooth muscle is going to divert blood towards the digestive system so that you can absorb that nutrients and then send the nutrients to the liver or the other parts of the body that need it. And again, if all the blood is going to be in my stomach absorbing the nutrients, it can't be in my skeletal muscles, so I'm not going to feel like going for a run or fighting somebody after I eat a big meal. Like we said, digestive glands are going to be activated while sweat glands are going to be deactivated. Adipose tissue, instead of burning through adipose tissue like we were in fight or flight, we're storing up adipose tissue now in this rest and digest. So it's interesting that it's like the same puppets that are being controlled by two different arms. One is for fight or flight. And you know how I mentioned in fight or flight, there are some receptors that are going to cause blood vessels to close up, like the ones that go to the digestive system. We don't want blood going to the digestive system if we're in fight or flight. That's no time to digest any food. Instead, we want blood going to places like the heart wall. So you'll see at the same time, some blood vessels are going to constrict while other blood vessels will dilate. And how they're able to do that is by these different types of receptors. We'll see that some are excitatory and cause blood vessels to dilate. Others are inhibitory and are going to cause those to constrict.
Can you spell out the NE and EPI?
Yes. I can also show you where all of that stuff is at. Did you get the class handout?
I did.
Yeah, I think you have to connect all of those adrenergic. Do I have epinephrine and norepinephrine on there? Maybe we don't. Oh, there it is. Norepinephrine or epinephrine. I just use the abbreviations for that.
43, 44.
Yep. I am always really crowded for space on here, so I'll tend to just use those abbreviations, and you're always welcome to use the same ones. I'll make sure to put those on the exam, just like the lecture handouts that are there. The point that I was trying to make, I guess, is that you could have, and I guess I'll just wait till we get closer to this, these adrenergic receptors are going to behave in different ways. Some are going to be excitatory, others are going to be inhibitory. I'll leave that, I guess, for more detail as we get closer to the effectors, but at least we've got them all listed on the board. The voluntary effectors, skeletal muscle, and then the involuntary effectors, everything else in that box. So maybe behind this screen is where we have all of the information processing, all of the central nervous system. We've got receptors on one side out here in the peripheral nervous system. We've got, again, central nervous system behind the screen, and then we've got out here in the other part of the peripheral nervous system all of the effectors, the motor division. Somewhere behind here, the information processing neurons that allow signals to go directly from receptors all the way over to effectors, these pre-programmed responses to a certain stimulus. This is called a reflex arc, a pre-programmed response to some type of stimulus. There are two of these reflex pathways that we'll want to introduce. I think there's something about this in the class handout. A reflex arc that involves some type of skeletal muscle is what we're going to call a somatic reflex. You may remember the motor division being separated into a somatic division and an autonomic division. Somatic goes to skeletal muscles. Autonomic reflexes are ones that go to cardiac muscle, smooth muscle, and glands. So things like those vital reflexes like sneezing, coughing, or vomiting, these are all autonomic reflexes and are going to involve that autopilot rather than the somatic division. Here's the thing that I'm talking about, where if we separate the right half of the board into this motor division, we've got one branch, the somatic division that I've got drawn in kind of this pinkish color. Skeletal muscle is the only thing we find at the end of this somatic division, but if we're going to find cardiac muscle, smooth muscle, or glands, that's going to take us into this autonomic division. And we've got the two settings there, sympathetic or parasympathetic, which we know is either fight or flight for sympathetic or rest and digest for parasympathetic. Also in the class handout, this is one of the first things that I want you to list the 4 structural components of the peripheral nervous system. And the first structural component are going to be these sensory receptors. We're going to briefly look at a few of the different types that we've listed. Sensory receptors are out in the peripheral nervous system. We also said that nerves and ganglia are part of the peripheral nervous system. Specifically, nerves are just bundles of those axon portions of neurons, and ganglia are going to be collections of neuron cell bodies out in the peripheral nervous system. The motor endings are all of these effectors that we see out here in the peripheral nervous system. And again, we've already seen the sketch that shows this, but these are the 4 things that you list under the very first item in our class handout, the 4 structural components of the peripheral nervous system. Sensory receptors, we can see these nerves, bundles of axon portions of neurons. We can see ganglia, these collection of cell bodies outside of the brain and spinal cord. And then those motor endings down here, either skeletal muscle or cardiac muscle, smooth muscle, and glands. Yes, what's up?
Oh, I just needed to read it. Sorry.
Oh, no problem. Here is where we kind of introduce special sensory versus general sensory. And we have, coming up, maybe next lecture, we'll be able to get into the special senses. And we're going to list these special sensory structures are all going to involve bipolar neurons. And special senses are going to be located just in the head. Whereas the first bullet point there, general senses, are body-wide sensory structures. So they're all over the body. They're not involved in things like vision or smell or taste or hearing or balance or equilibrium. Like all the special senses, those are all receptors we're going to find in the head. General senses are telling you things about temperature, pressure, these chemoreceptors. Whether you're stepping on a tack, we can take away photoreceptors and mechanoreceptors can stay. Thermoreceptors, again, are body-wide. I can feel if I'm touching something cold or hot. Chemoreceptors are body-wide. You have to be able to tell whether you are, your levels of oxygen or carbon dioxide are high or low in any particular tissue. And then pain receptors are also body-wide. So general senses are body-wide. They tend to involve unipolar neurons, while special senses are located in the head and tend to involve bipolar neurons.
But there's also general senses in the head.
Right. General senses are going to be everywhere. You can say special sensory are just in the head only. If we think about those sensory receptors themselves, we're going to zoom into them very quickly, and we're going to kind of classify them based on, either their stimulus, their location, or their structural complexity. The simplest of them are going to be those free nerve endings. We said those were simple. They were unencapsulated, just like the root hair nerve plexus. It was free nerve endings, unencapsulated. If we come over here to the sensory receptor, we're going to see that each sensory receptor, no matter what it's picking up, is going to be responsible for detecting stimulus over a certain given area. It's called a receptive field. Here's what one of those receptive fields look like. We can see these dendritic ends are kind of monitoring a patch of tissue. This is their receptive fields, respectively. These receptive fields, if hit with constant stimulus, can lose their sensitivity. This loss of sensitivity under constant stimulus is what we call adaptation. You experience this if you wear a watch or something, or wear a belt. You want, under that constant stimulus, you will forget sometimes that you're wearing a watch, or clothes are touching my body. You lose that sensitivity after a certain given amount of time. So that's what we call adaptation to these receptive fields. I said we would look at different receptor types, and like we said, different stimulus are going to be picked up by different types of receptors. And we can see in the picture some of these receptors are unencapsulated, like that root hair nerve plexus. We've got a free nerve ending that's unencapsulated versus the Meissner corpuscles or these Pacinian corpuscles, which are encapsulated by some layers of connective tissue. Again, mechanoreceptors like these hair cells are going to pick up directional force. Thermoreceptors are picking up temperature fluctuations. Photoreceptors are going to be picking up the sensitivity to light. Two other types, we've got chemoreceptors, which we said are monitoring chemical levels all over the body. pH, calcium levels, glucose, oxygen or carbon dioxide levels. And then nociceptors are body-wide, and they're pain receptors.
So the only one that is involved with special senses, photoreceptors?
We're going to see mechanoreceptors also involved in special senses, specifically in those hair cells, which I think I've plugged hair cell at least once, and we're going to get there with special senses. Instead of stimulus type, if we think about location type, we said exteroreceptors are located somewhere around the surface of the body. So if I'm stepping on a tack or slamming my hand in a car door, these touch receptors or temperature receptors, if I'm walking across a cold floor in the morning, these are all exteroreceptors, and I'm picking up sensations from the exterior of my body. One thing about those exteroreceptors, the sensory receptors that monitor patches of the surface of the body are laid down in these layers that are called dermatomes. They look something like this. I mean, there's slight fluctuations in these for any given individual, but these dermatomes, they map out this way in kind of this segmented fashion. And that's the main thing that we need to remember. There are these little receptive fields, these sensory patches that are monitored by certain sensory neurons. And these little patches we're going to refer to as dermatomes. And you can see they form these little segmented regions. One of my buddies the other day he was telling me that he had before, why is that? I don't know why that's slipping my brain for a second. Shingles. Shingles is what he had. Shingles. So shingles are basically these very painful blisters that can pop up on certain dermatomes, certain patches of the body. And what they basically have to do, if I go back a couple of slides to something right here, they have to do with sensory neurons that obviously are infected. And these sensory neurons that are basically sensing a patch of skin, if we follow that sensory neuron all the way out to the patch of skin that it innervates, these patches of skins are often in these dermatomes. And that's why you will see those shingle blisters, they'll kind of follow these dermatomes and can sometimes wrap around side of a body. Dermatomes is what we need to know. Responsible for these little sensory receptive fields on certain portions of the body. So we said those were exteroreceptors, these superficial receptors, and how they're spread across the surface of the body. We said interoreceptors are going to be in your internal vital organs. So if you have a stomach ache, or if there's some type of maybe a stretch in the lungs, or some type of intestinal pain, this is all going to be picked up by those interoceptors. Also called visceroreceptors because of the visceral organs. And then the third one, based off of its location, we said was proprioceptors, and they're found in the joints of the body, and they convey information about the position of those joints, whether they're in an okay, normal resting position, or if they're stretched in some kind of weird position. I woke up weird the other night because my arm, I guess I was laying on it in a weird position, and so those, again, the proprioceptors are sending in information that woke me up, told me to move around. So I'll tell you something about the position of your body's limbs. Finally, to the third box down here, looking at those receptors based off of their structural complexity. The complex ones we're going to see more when we get to special sensory. If we just compare the simple ones, they are either encapsulated in connective tissue, like we can see this Meissner corpuscle, or they are unencapsulated, like we would see for the free nerve endings or this root hair plexus. I believe that's it for receptors. I did add these, just a couple of diagrams that are more for reference. I wanted to make sure that we got the lecture handout questions correctly. And I think there's question number 13 on there that says all of these are classified as encapsulated nerve endings except, and if you click through here, you'll see that, I know that light is still kind of bright. Encapsulated ones, well, we've got this Meissner corpuscles are encapsulated. These Pacinian corpuscles are encapsulated. These structures called Ruffini endings are also encapsulated. What else do we have? Golgi tendons, these are encapsulated. They're right there embedded in this connective tissue. And then we have these muscle spindles, which we can also see are encapsulated in some type of connective tissue. So from the entire list there, the only ones on this list that are not encapsulated in some type of connective tissue are these hair follicle receptors, the nerve endings that go right there to the root of the hair. Not adipose, it's actually some of the dense regular connective tissue. So it is connective tissue, it's just not the adipose tissue. See if we missed anything there. You've got some matching about receptors based off of what type of stimulus they pick up or based off of their location. And we've answered the ones about encapsulated or unencapsulated. So where we are going next are going to get into, looks like some neurons and whether we consider them sensory or motor. So away from the receptor region and kind of into how do we get these, how this stimulus is converted into some type of electrical signal. Because neurons are going to have to be involved here to convert this stimulus into an electrical signal and then eventually get this electrical signal to bounce through neurons and control one of these effectors. So we'll see how this stimulus energy, different types of stimulus energy, is converted into what we will call receptor potentials, and how these receptor potentials, which are graded potentials by the way, are going to may or may not trigger some type of action potential. So in your brain, you already know action potentials are involving voltage-gated channels, and anything that is not an action potential is a type of graded potential. It's going to involve those chemically gated channels, these different types of receptors. Let's compare these different types of receptors. We're going to see as we look more closely, some of these receptors, like free nerve endings, for example, that's the dendritic end of a neuron. And like we said, what are located on the dendritic ends of neurons are going to be these non-specific ion channels. We have to remember what's going to trigger the opening or closing of these non-specific ion channels. In our previous examples, they were all some type of chemical signal. But again, sometimes they can be temperature, sometimes they can be mechanically opened or closed, sometimes they can be opened and closed based off of pain. So receptors, they can either be the dendritic ends of neurons, and look how, once we move away from the dendritic end, we know where the action potential begins because that's when we involve those first voltage-gated channels. Voltage gates and action potentials go together. Chemical gates and graded potentials go together. This is one type of receptor ending where the dendritic end of the neuron is the receptive ending. Again, think free nerve ending. Versus something like this, where you've got a completely different cell that is acting as the receptor. And this cell may or may not send signals to a sensory neuron. This is an example of a hair cell. We'll see that the hair cell is the receptor itself. And it's detached from the sensory neuron. So we'll call these receptor cells. There's our sensory neuron. In the previous slide, remember, there wasn't a separate receptor cell. It was just the dendritic end of this neuron, maybe located on the surface of my hand, telling if I'm touching something cold or hot or stepping on a tack. So here's our receptive cell. We'll see hair cells as an example. And there are things that can happen to a hair cell to either cause it to release neurotransmitter, get it all excited, or inhibit this hair cell and prevent it from releasing any neurotransmitter. And as we know, if it is releasing neurotransmitter, if that is sufficient amounts of neurotransmitter, it may cause this postsynaptic neuron here to generate an action potential. If it's insufficient amounts of chemical signal, then we never will trigger these voltage-gated channels on the axon hillock and involve an action potential. What else? I like this picture because it shows what I was just pointing out. This is a hair cell, and it's its own separate receptor. It may or may not be sending signals down to this afferent neuron. These are the sensory neurons that are waiting to receive signal from this hair cell. So sometimes the receptors are separate cells. In this case, we've got the Meissner corpuscle, where you can really see it's the neuron itself. It's that little dendritic end of the neuron that is actually picking up those vibrations. I also like this picture. I threw this one in here because it shows, again, this pathway where we go from sensory neurons that have to receive a signal, and the signal has to be sufficient enough here on the dendritic end that it initiates an action potential. Again, action potentials involve those voltage gates. They're going to run all the way down the axon to the axon terminal. And then here we can see one of these, what would have to happen as that electric signal hits the axon terminal. We know voltage-gated calcium channels open up, calcium ions move in, and then neurotransmitter moves out and lands on these dendrites or cell bodies of this integration neuron. We've got a graded potential running through the cell body and the dendrites, and if that graded potential is sufficient enough, boom, it's going to hit that axon hillock of this multipolar neuron. This would be an integration neuron. And again, if we involve voltage-gated channels, they run all the way down the axon to the axon hillock. Here, this voltage signal is going to be converted to a chemical signal again as those chemical signals release into the synapse and eventually dock to those receptors on the dendrites and cell body. Once again, we're involving chemical signals. This would be a graded potential that may or may not be sufficient enough to trigger an action potential. If it causes that first voltage-gated domino to fall here in the axon hillock region, now is when we start our action potential that ripples all the way down the axon to the axon terminal. So here we have our pathway from sensory neuron through integration neuron out to the motor neuron. Again, our focus are the types of receptors and how they initiate this first action potential. We've kind of talked about this pathway in Unit 4. The other part of this are these effectors, where these signals go, either to skeletal muscles or to these involuntarily controlled things like smooth muscle, cardiac muscle, and glands. Away from the brain and spinal cord, let's get back out into the peripheral nervous system. So once we are outside this brain and spinal cord, we're part of the peripheral nervous system. We had these dorsal roots and ventral roots that we mentioned last time. It's on this dorsal root that you see the dorsal root ganglion, and we know where the dorsal root and ventral root merge. This is our spinal nerve. So ganglia are a collection of cell bodies. Here's another collection of cell bodies that runs basically parallel to the vertebral column. These are called chain ganglia. They're sometimes called paravertebral ganglia because they parallel the vertebral column. Look right there. There's the dorsal root ganglion. That dorsal root ganglion is going to house neuron cell bodies that are part of the sensory pathway, that afferent pathway. These are going to be ganglion that are housing cell bodies, these motor neuron cell bodies. So this is the motor division. We'll point out ganglia, these collection of cell bodies, and then the nerves themselves, which are just the bundled axon portion of neurons. Here's a nerve. We can see it's really just bundles of those axon portions of neurons. I'm going to grab one of these neuron models. Not that it's going to show back up on the exam, but when we looked at this model, we said this gray tissue that's around the outside of this model is the endoneurium. It's the connective tissue that wraps around the outside of one individual neuron. You can see the Schwann cells that are just underneath this endoneurium. I don't know if I said endomysium. It's a neuron, so I should have said endoneurium. So again, it's just to the outside of those Schwann cells. But now as we look at this image, you can see that a bundle of axons, each with their own endoneurium, are found within a fascicle. So a fascicle inside a nerve is a bundle of axons. Each fascicle is protected by its own layer of connective tissue that we would call perineurium, just like the paramycium in muscles that was around a fascicle of muscle tissue. The fascicle of nerve tissue is protected by the perineurium, and then around the outside, the very outside of a nerve, is the epineurium, and it houses fascicles of neurons. I was about to point out the thing that I had been mentioning a couple of times when we were first doing our sketch of the spinal cord model, where we were talking about the dorsal roots are always carrying sensory information. So we said dorsal roots were afferent fibers only. They're going towards the brain and spinal cord. Where the ventral root was motor nerves only. It were only these efferent fibers carrying signals away from the brain and spinal cord. But then when we got out to the spinal nerves, we said that they were mixed. They had both afferent and efferent signals. It was a two-way street. Signals going towards the brain and spinal cord, and also signals going away from the brain and spinal cord. Thinking of those spinal nerves, this also comes from one of the questions in the handout. Looks like question 21. We're going to see that while there are 12 pairs of cranial nerves, there are going to be 31 pair of these spinal nerves. And I think it may want you to list somewhere in there. This is on question 23. List in order the spinal nerves by name and order. And here you have on the right-hand side of this slide those spinal nerves by name in order. The first 8 spinal nerves are called cervical nerves, and they're found in the cervical region, the neck region. Then there are 12 thoracic spinal nerves, 5 lumbar spinal nerves, and then 5 of these sacral spinal nerves. The last one is the coccygeal nerve. So you can just list those in order. That's question number 23, the spinal nerves by their name and number. Then underneath that, you'll see that there are 4 spinal plexuses that we have to list in order from superficial to inferior. And if I can switch to this slide, you can see the names are still there if you're listing those cranial nerves from top to bottom. But now on the left-hand side of this slide, I've answered question 24, which are those 4 spinal nerve plexuses. I'm going to zoom into each one. Let's zoom into the first one. Here's the cervical plexus. We can see these spinal nerves that as they leave, they end up merging with other spinal nerves, and they kind of form this little spaghetti bowl here of mixing nerves. This is a nerve plexus that is found in the neck, our cervical nerve plexus. So C1 to C4 is the cervical nerve plexus, the first 4 of those cervical spinal nerves. If we drop beneath that from C5 to T1, the first thoracic vertebrae, this is what we call the brachial nerve plexus. And look at how we've got 3 of these spinal nerves that are all going to merge, and we end up having 1, 2, 3, 4. We've got 5 different nerves that end up going down and controlling the muscles of the arm. You've got this merging of spinal nerves. This, once again, is a nerve plexus. And the benefit of these nerve plexuses is, let's say, one of these nerves gets damaged in a fight. Well, thanks to this nerve plexus, we now have alternate ways for these downstream neurons to get controlled. So plexuses allow these alternate routes for neuron control. We have one in the neck, the cervical plexus, one here in the arm, the brachial plexuses. And if we go down into the leg, there's one that goes from the anterior side of the leg and then one down the posterior side. The anterior side is going to be our lumbar plexus, so from L1 to L4. This is a nerve plexus right here in the groin region that's going to make its way down into the anterior part of the thigh. It's going to end up feeding into the larger nerves that we're going to call the femoral nerve and the obturator nerve. Again, femoral nerve along the anterior side. If you think about the nerve that's along the posterior side of the thigh, that's the sacral nerve plexus. So if somebody's complaining of sciatic nerve pain, or they're grabbing the hamstring, the back side of their thigh. So sacral nerve plexus. I don't know if you can tell, maybe the ankles tell that we're looking at the posterior side of this individual, and that's where this sciatic nerve runs. Again, it feeds the tibial nerve and the common fibular nerve. We've got through the nerve plexuses there. Again, we can see these spaghetti bowls, basically, where nerves are merging together and then branching back off. Allows, again, for multiple routes of signals to be sent to those neurons. But you have to know 12 pairs of cranial nerves. Here are the names of all 12 cranial nerves, and you've even got a couple of G-rated versions of these acronyms that you can use to help remember the names of those cranial nerves. You can see there are Roman numerals in front of each cranial nerve that indicate, what number cranial nerve it is, 1 through 12. So you can use these acronyms to just remember the names of the cranial nerves. We've got OOO, to touch and feel very green vegetables, or to touch and feel very good velvet. So the A and the H are the accessory nerve and this hypoglossal. You can come up with your own so that it allows you to remember the names of those cranial nerves. But in addition to knowing the names of the cranial nerves, we also need to know what those nerves do. So this will allow you to remember the names of those cranial nerves, but not necessarily what each of them do. Let me do this sketch. It's something that I've taken from the internet and I've modified it. So you may see various versions of this. But instead of trying to map which ones are sensory, which ones are motor, and which ones are both, I'm just going to try and draw with this one color these 12 cranial nerves. And this is something, there's always scratch paper available on these exams. So one good thing that it might be helpful to do is if you can take this and draw it on a blank sheet of paper right at the beginning of the exam, you can refer back to this diagram and it might help you navigate these 12 cranial nerves. And to start this, what we're going to do is we're going to take cranial nerve number 7, that facial nerve, and I'm just going to draw 2 number 7s facing one another. Hang in there with me. And then I'm just going to take this vestibulocochlear nerve, our cranial nerve number 8. That's going to be the ones that picks up hearing and also equilibrium and balance. That is going to be found in the ears. So I'm going to make the ears of this diagram number 8, just to represent the hearing, equilibrium, and balance. And you can see the 2 number 7s that are facing one another are going to form this boxy head that I'm trying to draw. And then right in the middle, the nose of this face, I'm going to put a number 1. That tells me olfactory nerve, the first cranial nerve. And this is found in the upper part of the nasal cavity, and it's for smell. So it's representing our nose on the diagram. Then for the eyeballs, I'm going to put 2 number 2s. The second cranial nerve is our optic nerve, and so that's the one that picks up images. So it represents our eyes. Then the third one, the oculomotor nerve, I'm going to draw that as a number 3 that's just kind of fallen on its face. So there's a number 3 that's just kind of fallen over, and it's kind of making up the eyebrows of this diagram. The oculomotor nerve, we're going to use this oculomotor nerve whenever your pupils constrict or dilate. Here's an example. Ooh, it just got darker, so now your pupils had to dilate to allow more light in to see what's on the board. Your eyes are able to constrict, your pupils are able to constrict or dilate due to the working of this oculomotor nerve. Hang on, because we're turning one of those back on. So now that oculomotor nerve had to work again to constrict the pupil to prevent as much light from getting in there. Okay, so ocular motor allows us to constrict or dilate our pupils. Then if you're following somebody, making them follow the light, if you're doing some kind of sensory test, if they're not able to move their eyes in an inferior direction, then there's problems with the trochlear nerve. So number 4, I'm going to put little number 4s kind of right underneath the eyes. This is like if you're, I see this sometimes with people driving on the road, they've got the phone in their lap, but they don't want to look like they're looking down at the phone. So their head is facing forward and their eyes are looking down or something like that. If you're looking at your notes, but your head isn't looking down, you're just pointing your eyes inferiorly, that's due to the functioning of this trochlear nerve. So if that is damaged, your eyes cannot move inferiorly. You'd have to tilt your head down. Okay, so trochlear nerve, that's number 4. The trigeminal nerve, this is cranial nerve number 5, and it's once again one of these large cranial nerves that's going to be involved in a lot of the facial expressions that you can make. And so what I'm going to do is I'm just going to put a big number 5 that kind of represent the face of this diagram. So number 5 and number 7 are the largest of these 2 cranial nerves that we've drawn so far. The next one on the list is number 6, and I'm going to put little number 6s just to the lateral sides of these eyes. Cranial nerve number 6 is our trochlear nerve. Sorry, let me try that again. Cranial nerve number 6 is our abducens. Trochlear nerve does control the eye, but it moves the eye inferiorly. Abducens abducts the eye. We said abduction is movement away from the midline, and the abducens is going to move the eye away from the midline of the body. So again, you're looking forward. I don't want to turn my head to see what's out that window, but I'm going to cause my eye to look over there. So lateral movement of the eye is done by this abducens. If you can't move the eye laterally, then you've damaged the abducens. So again, causing somebody to follow the end of your pen can sometimes tell if there's cranial nerve damage in a particular area. We've already got 7 on the board. That's the edges of our face. So this facial nerve is cranial nerve number 7. Vestibulocochlear nerve we have as our ears. Vestibulocochlear nerve is going to be what we talk about when we get into special sensory and talk about hearing, equilibrium, and balance. The next one is the glossopharyngeal. This is cranial nerve number 9. And here's what I can do. I'm going to put a tongue that sticks out. Notice how 2/3 of the tongue, this anterior 2/3 of the tongue, I'm going to put a number 7 there because your taste receptors on the anterior 2/3 of the tongue, that sensory perception is picked up by cranial nerve number 7, this facial nerve. However, the posterior 1/3 of the tongue, your taste receptors on the back of the tongue, that sensory reception is picked up by cranial nerve number 9, this glossopharyngeal. Now, vagus nerve number 10, this vagus nerve is a really long cranial nerve, and it actually wanders into the thoracic cavity and down into the abdominal cavity. It hits a bunch of digestive organs and the heart and the lungs. It wanders around to these different organs, and that's where it gets this name, vagus nerve. It's named after the word vagabond, for wandering nerve. And what I'm going to do to show that it wanders down into another organ, I'm just going to draw a heart down here. In the middle of the heart, I'm going to put a number 10 for this vagus nerve. I'll just say it this way. The heart would normally want to beat at about 100 beats per minute, but average heart rate is about 76 beats per minute because you have this constant slight signal being sent from the vagus nerve to the heart that controls the heart rate, slows it down. So 76 beats per minute is around normal. So our vagus nerve we've drawn here in the heart. Then number 11, this accessory nerve, I'm going to draw that here as the shoulders. I'm just going to put a stretched out number 11 over there, and I'll put another stretched out number 11 over here. And this accessory nerve is the one that you would use to shrug your shoulders. So it innervates that trapezius muscle. If you are not able to shrug your shoulders, then there's damage to this accessory nerve. And then the final one that I'll put on here, this is the only one that I'll just put in a different color. Because what we are about to do is we're about to, I'll list something over here that will signify whether these cranial nerves are just sensory, just afferent pathways, whether they are just motor pathways going away from the brain and spinal cord, or whether they're both. This last one is motor only. I'm going to put this in red just to kind of signify that this is a motor neuron. It's not picking up any sensation. You're not tasting with this at all. But this hypoglossal nerve is the one that controls some of the muscles of the tongue. So to move around the tongue requires a motor signal, and that's going to be done by this hypoglossal. So the tongue has both sensory and motor neurons that go to it for picking up taste, but then also controlling the movement of the tongue. Really, the rest of the slides just go through in once again, a slower fashion that point out each of the functions of these 12 cranial nerves. But I can promise you that you'll have to remember the names of these 12 cranial nerves, however you want to do it. G-rated acronyms are available for you. There will definitely be some questions, like you see on the class handout, about the functions of these cranial nerves. And then also in the class handout, we're about to get into this section where we need to know something about whether a nerve is purely sensory, whether it's purely motor, or whether it's a mix of both. And I didn't do a good job of color coordinating sensory, motor, or both. So let me try and do that now. Let's see if I can get it. I'm going to get a blue color for sensory. I'll get a red color for motor. And then I'll just use this black color for both. And I'll list, let's just list it this way. I'm going to list the 12 cranial nerves. 1, 2, 3, 6, 8, 9, 10. Okay, so we've got 12 cranial nerves, and if we want to know which ones are I'll just put it up here. Sensory. Red is going to be motor. And this color, I guess, the black color will be both. Both sensory and motor. So here's an acronym that you can use to list and quickly determine which of these cranial nerves are sensory, motor, or both. This also comes from the textbook. And the, let's see if I can do this right with the colors. The acronym, it's 12 letters, and it is some say, and I'll just do this with an S. So some say marry money. Let's see. Marry money. But, there's the B. But my brother says big brains matter most. Got all the way through it. So now, that's right. So we said, some say marry money, but my brother says big brains matter most. What this has done for us now is that we know the first two, the olfactory nerve and optic nerve, these are just sensory nerves that are picking up afferent signals, sending them to the brain and spinal cord. We said for ocular motor and for this trochlear nerve, these are both motor. They're just controlling the movement of the eye. In the case of the third one, this is controlling, we said, whether the pupil can constrict or dilate. Think about, coming out of the movie and it's really bright outside and your pupils have to constrict. That's this ocular motor nerve. Trochlear nerve is allowing you to move the eye in an inferior direction. Jumping to 6 is the abducens. It allows us to move the eye in a lateral fashion. We said the trigeminal and the facial, these are big cranial nerves, and they actually have a mix of functions. They have both sensory and motor functions. One of the things I should mention, the trigeminal nerve. This is one that allows you to feel sensation if somebody is touching your cornea. So on the list, you can see if you cannot feel sensation when somebody is touching your cornea, then there's damage to this trigeminal nerve. Facial nerve number 7, this is another one of these mixed nerves. It has both sensory and motor functions. One of the motor functions of the facial nerve is allowing you to blink your eye. So obviously if you are capable of blinking your eye and the blink reflex, facial nerve is fine. But if you can't blink your eye, somebody's touching your eye and it's not blinking, then there's damage to that facial nerve. Anyway, down the list we said number 8, this is all sensory. Sure enough, when we get to the ears, we'll talk about sensory information, hearing, equilibrium, and balance, all done with this vestibulocochlear nerve. Glossopharyngeal nerve, as well as the vagus nerve, are both mixed in terms of sensory and motor. And then we have just motor nerves. The accessory is the one that controls the trapezius, the shrugging of the shoulders. So if you can't shrug your shoulders, something wrong with this accessory nerve. Then hypoglossal is the one that allows you to move your tongue around. So if you can't move your tongue, then there's something wrong. If you can't stick your tongue out or protrude your tongue, if you have difficulty speaking or swallowing, then there's damage to that hypoglossal. Okay, went through a bunch of that. We've gone through these acronyms. You've got the one that allows you to remember the names of the 12 cranial nerves, as well as an acronym that will allow you to remember which ones are sensory, motor, or both. And this diagram that you won't ever have to draw, but it might be helpful because you might be able to answer some of the multiple choice questions about which cranial nerve is faulty if they're not able to do something like blink their eye or control pupil dilation or equilibrium or balance or protrude the tongue, whatever it may be. The rest of this PowerPoint really is just, once again, walking through those 12 pairs of cranial nerves. And these are slides that you can click through. We can see this olfactory nerve is found in the superior part of that nasal cavity. Its main function is sense of smell. So we have it as our nose up there on the diagram. These optic nerves, this is cranial nerve number 2, again picking up vision. We're going to end up drawing the cells of the retina to see exactly how it processes images, I guess in 2 lectures from now. So damage can cause blindness. Number 3, this ocular motor nerve, we said this is the one that really functions in constriction of the pupil. So allowing the pupil to dilate or constrict is something controlled by that ocular motor nerve. The trochlear nerve, we said, was what allowed you to move the eye inferiorly. So rotates the eye inferior and laterally. Trigeminal was the next one. This is a really large nerve. You can see it going to places all over the face. So we really stretched it out and it represented the entire face on our diagram. It's one of these mixed nerves. It's got a lot going on, sensory and motor function. We said one of the main things is allowing you to tell whether or not somebody is touching your cornea.
What did the ocular motor one do?
Ocular motor constriction of the pupil. Also opening of that eyelid. So we kind of have it up here as the eyelids in our diagram. So trochlear was inferior movement of the eye. Trigeminal was all of the facial expressions and also able to tell if somebody's touching your eye. So that sensation on the cornea. That's a functioning trigeminal. Trochlear, sorry, I went to trochlear already, the inferior movement of the eye. Now we're at abducens, so I've done that twice, where the abducens is abducting the eye, lateral movement of the eye. Then in number 7 is another one of these large facial nerves, or large cranial nerves. It's the facial nerve. You can see it going all over the sides of the face. It has both motor and sensory functions. We said taste on the anterior 2/3 of the tongue, we pointed out. And then we also said it helps control the blink reflex. So if you can feel somebody's touching your eye, then trigeminal nerve is fine. But if you can't blink, then something is off with this facial nerve. Then vestibulocochlear nerve. This is a sensory nerve that represents our ears. We're going to see there is the vestibulocochlear nerve. It's hooked up to this thing that looks like a snail that's got these little loops coming off of its head. We're going to talk about this snail structure that's got loops coming off of its head when we get into the ears. We'll see one part is picking up sounds, that's the snail shell, and then the other part that looks like a series of loops is picking up your equilibrium or balance. Number 9 is our glossopharyngeal. We said it picks up taste sensations on the posterior 1/3 of the tongue. Since it's also in the back of the throat, it's involved in swallowing as well. Then vagus nerve, this is the vagabond nerve. You can see it wandering into many of these other internal organs, digestive organs, heart and lungs. One of the things it's also involved in is it's involved in speech. So I think there's some lecture handout where it says if you've gone hoarse and you've lost your ability to speak, one of the things that could be damaged is this vagus nerve. Again, damage causes hoarseness or loss of voice. May also impair swallowing. The accessory nerve, we said, goes to these trapezius muscles and allows you to shrug your shoulders. So damage would prevent you from shrugging your shoulders. And then the last one is the hypoglossal that goes to those muscles of the tongue and allows you to protrude your tongue or manipulate food in the mouth. Right, and that takes us to the end of our peripheral nervous system. I'm going to have to get this screen out of the way at some point, but just to kind of show where we're going now. We just got done talking about the sensory division and some of the receptors that are picking up sensory information. And now what we're going to focus on, really the other half of the board, which is this motor division. How do we get signals out to those effectors? There in this motor division going away from the brain and spinal cord, we have one branch, which is the somatic division. That was what we drew here on top. Skeletal muscle. I'll write this over here on the other hand. I'll put... Let's get this out of the way. I'm going to put one line. On one side of this line, it's going to represent the central nervous system. So this is going to be brain and spinal cord. Everything on this side of the line is going to be the peripheral nervous system. So we've gone outside of the brain and spinal cord. And the peripheral nervous system is eventually going to lead out to these effectors. The top of this, we're putting what we're going to call the somatic division. Maybe I'll just try and, the best I can, try and separate this somatic division, which is just referring to skeletal muscle, and then beneath that we're going to write our autonomic division. I'm going to try and draw these two. Autonomic is the autopilot. We know it operates in what we call sympathetic or parasympathetic, so we're going to draw those two pathways. I'll start by drawing this somatic pathway first. In a somatic pathway, what we're going to see is one motor neuron. And these motor neurons, these are under voluntary control. What else do I want to say about these? We're going to see the neurons of this somatic division. Their cell bodies originate in the anterior gray horn. So we got one motor neuron. Let's draw what one of those motor neurons is going to look like. I'll try and draw it in this same, I'll just try and keep this same color. We know all motor neurons are multipolar. So I'm going to draw this multipolar neuron. The cell body and the dendrites are located in the central nervous system, specifically this anterior gray horn. And then we're going to see the axon portion leaves and goes out of that ventral root. And it's going to make its way all the way. There's just one of these motor neurons. So this long axon is going to come all the way down right to the motor end plate. I'm just going to draw this expanded axon terminal. We know there's going to be vessels of neurotransmitter in there, that acetylcholine. And with this one motor neuron, we're going to see that it is going to have heavy layers of myelin. So I'm going to draw these really thick Schwann cells all down the length of this cell. I can list that here, heavy myelin. So that means these signals that travel down this somatic branch can travel very quickly. We've got just one motor neuron. They're voluntarily sent. So again, if you want to control one of those skeletal muscles and cause your big toe to wiggle, you can voluntarily decide to do that. We're going to compare this pathway, this very simple one motor neuron that's under voluntary control from the anterior gray horn out to whatever skeletal muscle it goes to these pathways of the autonomic division. I know I have put the slides away. Let me get two colors, maybe this one and this one. I'll take that back. Maybe this one. There we go. In the autonomic branch, we know this is all the autopilot. We're under involuntary control, but there's some differences when we look at the anatomy of the autonomic nervous system. Instead of just one motor neuron, we're going to see two motor neurons. With two motor neurons, there is now going to be a presynaptic neuron and then a postsynaptic neuron, right? That synapse is that little gap in between two neurons that talk. So we have a presynaptic and a postsynaptic neuron. These are under involuntary control. In addition to being involuntarily controlled, we're going to see the cell bodies of these autonomic neurons are located in the lateral gray horns. I'm going to say brain stem or lateral gray horns. If I can spell it. Lateral gray horn. So we've got two motor neurons, and hopefully I've left myself enough space to draw these two things. The autonomic nervous system, let me show the two settings of this. In one setting, I'll just put this green. Actually, I'll try and stay color coordinated with what I've done over there. So far in a blue color, I'm writing the parasympathetic stimulus. So let me draw the parasympathetic neurons in a blue color. So parasympathetic division. This parasympathetic division is sometimes called the craniosacral division. We're going to see, because the cell bodies are located either in the brainstem, the cranial section, or all the way down in the lower part of the vertebral column, what we call the sacral region. And I'll draw one of the cell bodies. If we're thinking of these motor neurons of the parasympathetic nervous system, we're going to see that they have there's two motor neurons here. The first one is going to have a very long axon. It's going to leave the brain and spinal cord and make its way out here in the peripheral nervous system. It's got this long preganglionic neuron. I'm going to come all the way out here, and then this presynaptic neuron is going to come to an end. So we've got a really long presynaptic neuron, and here's our postsynaptic neuron. It's got a relatively short axon. I'm going to make it a little bit longer just so I can label it. Now that we've got two neurons, we can label this one our preganglionic neuron, and here's our postganglionic neuron. What's it, postganglionic? Preganglionic neuron. And this structure right here, let's just go ahead and put a dotted line. We're going to see a collection of neuron cell bodies outside of the brain and spinal cord. What did we call a collection of neuron cell bodies? Well, the name is already right there, so I guess I won't make that too much of a question. Neuron cell bodies outside of the brain and spinal cord, we said, are ganglions. And this collection of ganglions that we see as part of the parasympathetic nervous system, we're going to call these terminal ganglia. Basically because they terminate right at the end of this postganglionic neuron. They terminate very close to the effectors that they're going to control. Okay, so in the parasympathetic division, we've got a long preganglionic neuron and a short postganglionic neuron. Here's another thing that we can add. When we're thinking about the autonomic nervous system, we're going to see it's only the preganglionic neuron that's going to have any layers of myelin whatsoever. And I'm trying to draw these layers of myelin very thin. So we can say it has very light layers of myelin on the preganglionic neurons. The postganglionic neurons, they have no myelin whatsoever. So I'll just say no myelin. Okay. I don't know if we want to do this yet, but are we doing okay so far? We've got the structures of them down. There's only one thing I want to add to it.
I had a question.
Sure.
On the top one, how the axon terminal is a circle, is there a difference on why the bottom one is just like a little?
I'm lazy. Let's draw that one as a circle too. There we go. It's just in those axon terminals, that's where you find those vessels of neurotransmitters. And that's exactly where I'm going. So let's go ahead and make sure we're drawing those axons with those axon terminals filled with little vessels of neurotransmitters correctly, because we're about to add which neurotransmitter is in there. And hang in there with me, because so far the only neurotransmitter that we've talked about, the only chemical signal that's coming out of the ends of these neurons has been acetylcholine. That's the only thing we've talked about so far. We're going to see acetylcholine comes out of the ends of these somatic neurons. It's also going to come out of the ends of the preganglionic and postganglionic neurons when we're talking about the parasympathetic nervous system. It only ever changes when we get down to the sympathetic nervous system. Only in the sympathetic nervous system are we going to see these other chemical signals, epinephrine and norepinephrine. Those are going to start to ramp everything up. So hang in there. We're going to see epinephrine and norepinephrine when we talk about sympathetic division, fight or flight. Right now, we're still in parasympathetic division, this rest and digest. And in parasympathetic stimulus world, we're going to see that both the preganglionic neuron, if we go to the end of this neuron, we're going to see the vessels at the end of the preganglionic neuron are filled with acetylcholine. And it's acetylcholine that's released into this synaptic cleft. And it has to dock to those receptors on the dendrites and cell body, the same receptors that we were talking about last time. They are sensitive to acetylcholine, and if enough acetylcholine is docking to those receptors, they open. They allow sodium to rush into this cell body. If enough sodium rushes in, then we might trigger an action potential along the axon portion. So the neurotransmitters that comes out of the preganglionic neuron is acetylcholine, and even the neurotransmitter that comes out of the postganglionic neuron is also acetylcholine. So postganglionic neurons that release acetylcholine belong to the parasympathetic nervous system. And parasympathetic signals, here they are. If we follow this acetylcholine out, I can just draw it coming directly over here. Acetylcholine is going to be able to dock to these muscarinic receptors on cardiac muscle, smooth muscle, and glands. Under parasympathetic stimulus, we know what's going to happen to these cardiac muscles, to these smooth muscles and these glands. When these blue receptors are activated, we're resting on the couch. Our heart rate slows down. Smooth muscle diverts blood to the digestive system and away from skeletal muscles, activates the digestive glands and inactivates sweat glands. Adipose tissue, we are storing adipose tissue if we are in rest and digest.
Sorry, I got lost. Where did those receptors come from? Those are like receptors on dendrites?
These are receptors that are located on target tissues. These are receptors located on things like cardiac muscle, smooth muscle, and glands. Like we said, if you want to wiggle your big toe, you can control skeletal muscle, but you can't decide to control things like cardiac muscle, smooth muscle, or gland activity. All of this stuff is controlled by the autopilot, the autonomic nervous system.
So we just have those muscarinic receptors sitting on all those cardiac muscles, smooth muscles, all those things, and when the acetylcholine is released, it binds to those?
That's right.
Okay.
Muscarinic receptors is named after muscarine, which is that was a first, I think, found in mushrooms. And if you've, I don't have a lot of experience with that, but if you've, yeah, there you go. But if you see, like, what happens is like salivary glands get activated. If muscarinic receptors are stimulated, you'll see heart rate slow down. You'll see glands, like digestive glands, become activated. That's where you were getting into the question about medications. Certain medications that are going to trigger muscarinic receptors are going to have an effect, a parasympathetic effect, to slow down heart rate, to increase digestion. And other drugs might have a sympathetic effect that's going to target those exact same things, but in a completely different way, because we're triggering a different set of receptors.
It's still like new.
That's fine, but as it bounces around in there, throw them out. That's good to think about. Nicotinic?
Nicotinic receptor. So where they are, is they going to be in the motor or in this?
Great question. That's one of the last details. If I can get to the fact that these nicotinic receptors are going to be located in 3 different places. Nicotinic receptors are a type of cholinergic receptor. Cholinergic receptors are a class of receptors, anything that binds acetylcholine. If we see acetylcholine docking to a receptor, then it's a cholinergic receptor. Anatomically, cholinergic receptors, let me say it this way. There are two types of cholinergic receptors, either the ones located on cardiac muscle, smooth muscle, and glands. Those receptors that bind acetylcholine and are located on cardiac muscle, smooth muscle, and glands, we call those muscarinic receptors. And there's another class of cholinergic receptors that also binds acetylcholine, but it's not located on cardiac muscle, smooth muscle, and glands. The other group are called nicotinic receptors, and they're located in 3 places. One place is on motor end plates of skeletal muscles, and we've drawn those. We've also drawn the second one. These are also nicotinic receptors. Nicotinic receptors. They're located on postganglionic cell bodies and dendrites. So we've got skeletal muscle motor end plates, we've got postganglionic dendrites and cell bodies, and there's one other place that we find nicotinic receptors. I'll give it away. It's the adrenal medulla. We're going to draw that in just a second. But the adrenal medulla is involving epinephrine and norepinephrine, so to do that we'll have to draw sympathetic stimulus real quick. Let's compare sympathetic division to what we already have up here, this parasympathetic division. And I'll try and stay color coordinated. So I'll take my bright red and we're going to draw this sympathetic division. If we look at this sympathetic division, it's sometimes referred to as the thoracolumbar division because we're going to see the neuron cell bodies originate either in the thoracic region or the lumbar region of the vertebral column. So thoracolumbar refers to this sympathetic division. Once again, we've got 2 motor neurons. Let's draw the first motor neuron. I'm going to try and fit 2 different possibilities for sympathetic division. I'm going to draw my first motor neuron. We've got the cell body located here in the lateral gray horns of the thoracic or lumbar region of the spinal cord. We've got the axon making its way out into the peripheral nervous system. And if we're looking at sympathetic neurons, we're going to see that the first neuron, this preganglionic neuron, is really short. I'll just put it coming to a dead end, not too far away from the brain and spinal cord. And then we'll see a second motor neuron that it docks to. This second motor neuron is going to have a really long axon. Where this second axon is going to dead end close to those effectors, cardiac muscle, smooth muscle, and glands. Our preganglionic neuron is going to have very light layers of myelin. And our postganglionic neuron, once again, has no myelin. So we've labeled that. Look what we have. We've got a cell body that's outside of the brain and spinal cord. This neuron cell body that's out in the peripheral nervous system, we know we're going to call it a ganglia. This is actually part of what's given 2 different names. We can call this the chain ganglia. Or another name for this is the paravertebral ganglia. Paravertebral because this is actually a chain of ganglia. If I were to go down a little bit, we would see another ganglia below. If I were to go down even further than that, we would see yet another one of these ganglia. So what I'm attempting to draw here with my little series of circles is this part of the spinal model where if you look at these vertical structures, you can see these sympathetic chain ganglia, these vertical ganglia that parallel the vertebral column. So that's basically what I'm attempting to draw. I want that to get in the way. I'm attempting to draw this chain ganglia. And we're going to see that in the sympathetic division, the preganglionic neuron is very short, and it more often than not will stop at the chain ganglia, and that's where the second motor neuron will pick up and go all the way out to the effector. Before we add the second pathway here, let's add the neurotransmitters, because this is the only time we've switched the story up so far. In sympathetic division, once again, the very first synapse that we come across is going to be between a presynaptic neuron and our postsynaptic neuron. It's always going to be the case that the presynaptic neurons, whether it's sympathetic or parasympathetic, are always going to be releasing acetylcholine. So if you see acetylcholine as a neurotransmitter, that acetylcholine has to be docking to a receptor for acetylcholine. These acetylcholine receptors that are located on postganglionic neurons in cell bodies, we call those nicotinic receptors. So once again, these are also nicotinic. They're docking acetylcholine, and they're located on neuron cell bodies and dendrites, postganglionic neuron cell bodies and dendrites. So the preganglionic neuron is always releasing acetylcholine. The second motor neuron in this case is where the story changes. Now in the sympathetic division, we're going to see the postganglionic neuron switches it up, and for the first time we see a different neurotransmitter. The neurotransmitter that comes directly from postganglionic neurons, this is going to be norepinephrine. I'll just abbreviate this as NE or norepinephrine. It's basically the adrenaline molecule. It's going to dock to these adrenergic receptors and have an overall effect of increasing heart rate, increasing respiratory rate, burning through adipose tissue. There's another pathway for the sympathetic division, and it looks something like this. This is kind of this is less common in the sympathetic division, but we're going to once again see our preganglionic neuron cell body located in the lateral gray horns of the thoracic or lumbar region. Typically, in a sympathetic pathway, like we said, the preganglionic neuron is short and the postganglionic neuron is long. That's not always the case. There are some exceptions in the sympathetic division where this preganglionic neuron is going to come right through this chain ganglia and bypass it altogether. Just go straight past it, and instead this neuron is going to come out here and dead end into some type of organ. The organ that we're going to mention right now is the adrenal gland. So let me put a little double layered pyramid. This represents our adrenal gland. There is what we call this adrenal cortex, the outer edge of the adrenal gland, and then there's this middle medulla region of the adrenal gland. It's the medulla region of the adrenal gland that the preganglionic neuron goes directly into, and it dead ends right there in the adrenal medulla. Where's my blue color? So just like we had been putting before, the preganglionic neuron has a very light layer of myelin on it. It dead ends directly into, in this case, an organ, the adrenal medulla. Here's the blue color. Our preganglionic neuron, like we've been drawing all along, is always going to be releasing acetylcholine. So I'm just going to try and draw the release of acetylcholine into this adrenal medulla. There are cells in this adrenal medulla. I'm just going to put them as these little rounded cells. Those cells of the adrenal medulla are actually modified postganglionic neurons. So we've got this really long postganglionic neuron. In embryonic development, some of these postganglionic neurons became spherical cells embedded in the adrenal medulla. And just like normal postganglionic neurons, they have these tiny little receptors for acetylcholine. So they behave just like a postganglionic neuron. Those receptors for acetylcholine, if acetylcholine lands on these adrenal medulla cells, it's going to get these adrenal medulla cells, which are modified postganglionic neurons, to turn around and start to spit out neurotransmitters. These postganglionic neurons are going to be spitting out a mix of norepinephrine and epinephrine. Not that you really have to know this, but it's going to be about 80% epinephrine and about 20% norepinephrine. The point that I'm trying to make is that these are both adrenaline molecules. You can think of epinephrine as kind of like a cousin molecule to norepinephrine. Both of them are going to dock to adrenergic receptors. Both of them are going to have this sympathetic fight or flight response. But we're going to see some of these different types of adrenergic receptors are going to be more sensitive to one type or the other. For example, beta 2 receptors, these are like 100 times more sensitive to epinephrine than they are to norepinephrine. Other ones are going to be more sensitive. They would prefer norepinephrine than epinephrine. We don't have to know them that distinct from one another. We just know that epinephrine and norepinephrine are always coming out of postganglionic neurons in the sympathetic division. And epinephrine and norepinephrine, if we can draw these neurotransmitters making their way across, maybe make that a little better. Norepinephrine, and then we've got our epinephrine making its way to those same targets. We can see it's going to be able to control the cardiac muscle, smooth muscle, and glands in a completely different way. Let's add one other term. So we've got terminal ganglia that refer to these ganglia of the parasympathetic division. We've got chain ganglia or paravertebral ganglia because they parallel the vertebral column. And then we have in the sympathetic division, some of these ganglia, these are neuron cell bodies outside of the brain and spinal cord, but they're not part of this chain ganglia. I'll just squeeze this term in here somewhere. These are what we call prevertebral ganglia. Those are the prevertebral ganglia. They're the modified postganglionic neurons found in that adrenal medulla. We know the term ganglia just refers to these collection of neuron cell bodies out here in the peripheral nervous system. We've got terminal ganglia in the parasympathetic division. We've got both paravertebral and prevertebral ganglia as part of the sympathetic division. Let me see if there is the only other thing that I need to add. We said that adrenal medulla is the last place where we find those nicotinic receptors. So on this entire diagram, we know anywhere where we see these acetylcholine molecules docking. These acetylcholine molecules are always docking to some type of cholinergic receptor. Muscarinic receptors are the ones that are located on cardiac muscle, smooth muscle, and glands. They dock acetylcholine. The other places we see acetylcholine docking are on these nicotinic receptors. There's 3 locations for those: skeletal muscle motor end plates, adrenal medulla, and any of these postganglionic cell bodies and dendrites. Luckily, there's only one place where we find these adrenergic receptors, and it's on these same target organs that you find muscarinic receptors on. So this is where those same target organs, like we said, can behave totally differently depending on whether they are being hit with acetylcholine and having this muscarinic receptors activated, think rest and digest, versus if epinephrine and norepinephrine is bathing over these organs and adrenergic receptors being activated. We'll see this has the sum effect of the fight or flight. Effectors, where we started, we know the somatic nervous system is always going to these skeletal muscles. And in the somatic division, we said there's just one of these motor neurons, and it's always going down to the skeletal muscles. If we're thinking about the autonomic nervous system, we drew our autonomic neurons. They were always going over to the same effectors, that box that I put up that I just labeled as cardiac muscle, smooth muscle, glands like digestive glands or sweat glands, and then adipose tissue. We're going to focus on this autonomic branch. And we said the autopilot here has the two settings. One of them is the fight or flight setting, which is our sympathetic division. Then we've got our rest and digest division. What I like about this picture is that it shows the organs in the middle. These are, we can see smooth muscle, we can see cardiac muscle, we can see digestive glands. These organs are all dually innervated. So this is the dual innervation that I was trying to draw, that you've got the same group of organs being controlled at the same time by sympathetic signals as well as these parasympathetic signals. So dual innervation. What I also like about this image is if you look at where the cell bodies of these neurons are for the sympathetic division, they're really just coming off of the thoracic and lumbar region of the spinal cord. You don't find any of these sympathetic neurons down here in the sacral region or up here in the brainstem region. As opposed to if we come to this side, we can see that the parasympathetic division, those neuron cell bodies are originating up here in the brainstem and also down here in the sacral region. There's really none of these neuron cell bodies of the parasympathetic division that are found in the thoracic or lumbar region. So anatomically, these two divisions are located in different parts of the spinal cord. Again, if we think craniosacral, that's telling us parasympathetic. If we think thoracolumbar, that's telling us sympathetic. I knew at some point I would mix my colors up, but at least it's labeled correctly. We said this over. Sympathetic division is the fight or flight. So it's going to cause some effect of increasing heart rate, increasing respiratory rate, inhibiting digestion because it's no time for digesting food if you're trying to eliminate a threat or get away from a threat. And then parasympathetic stimulus was the opposite. It was the rest and digest setting. So it decreased heart rate, decreased respiratory rate. It allowed for digestion to occur. So think of after eating a big meal, you're reading a book on the couch and falling asleep. That's a parasympathetic signal. Now, since we're about to start to compare sympathetic and parasympathetic signals, I thought I would throw one little noteworthy thing out there. If we're thinking about sympathetic versus parasympathetic, one thing that we can give solely to the sympathetic division, and that is control of whether your blood vessels constrict or dilate. So this is a picture of a normal blood vessel. You can see there is a slight contraction to the wall of this blood vessel. It's partially constricted. And what can happen, this normal blood vessel that has a partial contraction of the muscle, we can actually take that partial contraction and relax it. So vasodilation would cause that slight smooth muscle contraction in the walls of most blood vessels to relax a little bit. And that vasodilation causes the diameter of the blood vessel to increase, so we're going to increase blood flow through the vessel if it's a larger diameter. If we wanted to decrease blood flow, then the opposite would be true. Instead of vasodilation, we would want to constrict that vessel. And what constricts a blood vessel is an increased sympathetic stimulus. So let me say that one more time. If we're trying to put something that is noteworthy for the sympathetic stimulus, we're going to see that the sympathetic division is solely responsible for controlling most of your blood vessels. In fact, the only blood vessels that are not controlled by the sympathetic division are the blood vessels that go to erectile tissues in males and females. In the erectile tissues of males and females, it's the parasympathetic stimulus that controls whether those blood vessels are constricted or dilated. That's the exception. So in every other blood vessel in the body, those are controlled by sympathetic division. And you can think of it this way. Sympathetic division has the volume turned up a little bit, and that's why we have a slight constriction in the walls of all blood vessels. And we can turn that volume, that sympathetic signal up louder, and if we increase that sympathetic signal, it's going to cause the blood vessels to constrict even further. Or we can dial down that sympathetic signal, and decreasing that sympathetic signal is going to cause the blood vessels to dilate. Most blood vessels are controlled entirely by sympathetic division. Increasing that sympathetic signal causes vasoconstriction. Decreasing that sympathetic signal causes vasodilation. It's not that sympathetic causes vasoconstriction and parasympathetic causes vasodilation. It's all sympathetic division. It either increases its strength and causes constriction or decreases the strength of sympathetic signal and causes vasodilation. Okay, so that's sympathetic tone, this fact that there is a constant sympathetic signal that's being sent to the smooth muscles on the walls of most blood vessels. I had already given previously, we said there was the fact that the vagus nerve sent a parasympathetic signal to the heart, and it caused the heart to beat at about 76 beats per minute, when on its own, without this constant parasympathetic signal, the heart would want to beat at about 100 beats per minute. So parasympathetic tone, an example of that constant slight signal, is the decreased heart rate. And we said blood vessel constriction or dilation for most of those blood vessels is going to be entirely controlled by sympathetic division. Okay, so we're looking at this picture now, and without even reading any of the things on the diagram, we can see we've got neurons that are originating either in the brainstem or down here in the sacral region. And if we look closely, you can also see that the first neuron, the darker blue neuron, our preganglionic neuron, is long compared to the postganglionic neuron that is relatively short. I don't know if you can see, there's a long preganglionic neuron, and then right there in the lungs is a very short postganglionic neuron. Here in the liver, there are very short postganglionic neurons. In the stomach and in the pancreas and the digestive glands, in all of the urinary structures, we can see long preganglionic neurons and a short postganglionic neuron. This is structurally indicative of our parasympathetic nervous system. We said craniosacral division, brainstem and sacral region. Once again, tells us parasympathetic nervous signals. If we're thinking about parasympathetic nervous system, we said rest and digest. So some of this is a little bit of a repeat. In addition to slowing down heart rate and slowing down respiratory rate, we said the digestive activity increases. Think again of digesting a big meal, reading a book on the couch after eating a big meal. What helps with that is if you're reading a book, on the couch, you're just looking at what's right in front of you, so that's going to cause pupil constriction. If instead you were trying to take in all of the stuff around us, your pupils would need to dilate to take in all of the information. Pupils dilating is what happens when you're in fight or flight, because you're thinking, okay, what are my outs here? Am I going to get out of this, or am I going to have to engage a threat? Sympathetic signals cause pupil dilation, and parasympathetic signal, this rest and digest, is going to cause pupils to constrict slightly. So this is accommodation for close vision. You're just reading a book right in front of you on the couch. Once again, this is just pictures of our craniosacral division, our parasympathetic division, long preganglionic neurons and short postganglionic neurons. We compare that to this sympathetic division, which we can tell this is sympathetic just by looking at it. We can see there is, well, first of all, there are these relatively short preganglionic neurons, and we see these really long postganglionic neurons. A lot of these postganglionic neurons are coming off of this chain ganglia, what we call the sympathetic chain ganglia, or we call this the paravertebral ganglia because these parallel the vertebral column. In addition to these paravertebral ganglia, you can see the few exceptions where there are collections of sympathetic ganglia that are in front of this paravertebral ganglia. These would be the prevertebral ganglia, sometimes called collateral ganglia or prevertebral ganglia. The one that we drew one of these preganglionic neurons that went directly through the chain ganglia. It passed all of the ganglia and went directly into that adrenal medulla, where it dead-ended into that adrenal medulla. What do we know about sympathetic stimulus? We've said it's fight or flight, so it's going to cause heart rate to increase, respiratory rate to increase. Your liver is going to start to burn through glucose. You're having to do information processing very quickly. We want to increase blood flow to the brain and decrease blood flow to digestive organs. No time to digest or absorb nutrients. We need that blood to go into the heart wall and into skeletal muscles so that we can get away from a threat or eliminate a threat, whatever we're having to do. Here's another, once again, that picture of that sympathetic chain ganglia and a few of these prevertebral ganglia. Typically short preganglionic neurons and longer postganglionic neurons when we're looking at the sympathetic division. Let's zoom in. What we've got here is a cross-section through the spinal cord. And just like we were labeling, you can see the preganglionic neuron. Its cell body is originating over here in the lateral gray horn. So coming off of this lateral gray horn and then out of the ventral root, we can see this motor neuron spin around, it completely bypasses that chain ganglia. This was the bottom neuron that I was drawing. I was drawing it bypassing that chain ganglia and going directly out into those cells of the adrenal medulla. We said those yellow cells of the adrenal medulla, those are modified postganglionic neurons. So they have nicotinic receptors on them that are binding this acetylcholine coming from the preganglionic neuron. Once they bind acetylcholine, their response is to turn around and start spitting out epinephrine and norepinephrine. We said about 80% epinephrine and about 20% norepinephrine. These are chemical signals that jump straight into the bloodstream and are very quickly into your bloodstream. Like this happens to me if traffic just causes it for me. But imagine, if we hear some kind of loud noise down the hall and it's some stressful loud noise, there is a neuron that directly goes to the adrenal medulla and in an instant can get this epinephrine and norepinephrine to be released into the bloodstream. So this is on the matter of seconds that a neuron can send a signal to get epinephrine or norepinephrine released into the bloodstream. So immediately your heart rate increases, your respiratory rate increases, you're instantly stressed out. Think about if we heard some loud noises down the hall. We don't know if it's gunshots or what. Our heart rate would all increase instantly. So the fact that you can get these chemical signals into the bloodstream very quickly is beneficial. We have to respond to threats very quickly. However, the downside is that there's no quick way to get these stress hormones out of the bloodstream. It's going to take minutes, sometimes an hour, for these stress hormones to wash themselves out. So it's very easy to get stressed, but not as easy to get unstressed. Here is a chart this is a page from the textbook, also the lecture slides. And all I've done is I've tried to draw it on the board. Let's see if I've left anything out. So we can see the color scheme is a little bit different. But what we wanted to do, we wanted to compare the somatic division to this autonomic division. And we said somatic division, there was always one motor neuron that went directly to skeletal muscles. This was the path that we were voluntarily in control of. But the other path, this is the autopilot. That autopilot, we said, has two settings, either the sympathetic setting or parasympathetic setting, and both settings are going to control the same effectors. So this is basically the box that I drew where I have cardiac muscle, smooth muscle, and glands and adipose tissue all in the same box. What else? So if we go back over here, we can see in the sympathetic division there's only one motor neuron and acetylcholine is released at the end of this motor neuron. We know it's going to dock to these motor end plates. If we get into the parasympathetic division, let's jump down to the autonomic division. I'll start with the parasympathetic first because that's what I just said. Remember, in the parasympathetic division, it's all acetylcholine. The preganglionic neuron is releasing acetylcholine and the postganglionic neuron is releasing acetylcholine. It's all acetylcholine in the parasympathetic division, and it's only half acetylcholine in the sympathetic division. Remember, the preganglionic neuron is always releasing acetylcholine, so it's the most common, but it's the postganglionic neurons. Here's one postganglionic neuron, and then down here there are modified postganglionic neurons. But all postganglionic neurons behave in the same way. If they are hit with acetylcholine by the preganglionic neuron, they turn around and spit out either epinephrine or norepinephrine. And it's that epinephrine or norepinephrine that causes the excitatory fight or flight response from these same target organs. So what I want to add to this are muscarinic receptors, nicotinic receptors, and then adrenergic receptors. Maybe we just did it there. Remember, nicotinic receptors are found in 3 places. Nicotinic receptors, I tried to put them in a blue color because this signifies they are they belong to the cholinergic class of receptors. Cholinergic means they dock acetylcholine, and we see acetylcholine docking in a few places. The acetylcholine that docks to cardiac muscle, smooth muscle, and glands, those acetylcholine molecules that dock to these target organs are going to dock by way of muscarinic receptors. If acetylcholine is instead docking to the adrenal medulla, postganglionic cell bodies, or skeletal muscles, these are all nicotinic receptors. So nicotinic and muscarinic are all docking acetylcholine. The difference is their physical location. One is located, again, on cardiac muscle, smooth muscle, and glands. The others are located on postganglionic cell bodies, adrenal medulla, and skeletal muscles. Adrenergic receptors are only located, again, on cardiac muscle, smooth muscle, and glands, and they can cause that fight or flight stimulus. I really like this slide. In fact, that's the thing that we tried to draw. I think we added all of that to our diagram. So I'll skip over it briefly just to actually zoom in to these different parts. Cholinergic receptors are always, anything that docks acetylcholine is a cholinergic receptor. Anything that docks epinephrine or norepinephrine is an adrenergic receptor. Let's take a second and look at this slide. There's a lot of detail on this, but I wanted to add this one. Maybe you can put a star next to this slide. We're going to do some case studies next time that it'll be helpful to have this slide handy whenever we do the case study. So we'll certainly get to spend a lot more time with this slide on whatever the next day is, Tuesday. But here's what we've covered so far. Cholinergic fibers, we know these are the axons that are releasing acetylcholine. Adrenergic fibers, we know that these are the axons that are releasing epinephrine or norepinephrine. We know that. So the fibers are the parts of the neurons that are producing and sending those chemical signals out to their axon terminals. The adrenergic receptors are, we'll start at the bottom. Adrenergic receptors, I've drawn them here. They're located on the target organs, in this case, our cardiac muscle, smooth muscle, and glands. Then there are cholinergic receptors as well, and these cholinergic receptors I've drawn in blue, either a dark blue or a light blue. So we can see any receptor that docks acetylcholine is a cholinergic receptor. And like we were trying to draw on there are two classes of cholinergic receptors. The nicotinic receptors that are found in 3 places. We said motor end plate of skeletal muscles, postganglionic cell bodies and dendrites, and that adrenal medulla. Whereas muscarinic receptors, those are the receptors always located again on cardiac muscle, smooth muscle, and glands. You see that muscarinic receptors and adrenergic receptors, they have the same docking sites, cardiac muscle, smooth muscle, and glands. One of them is rest and digest. The other receptors are going to trigger fight or flight. So this is that dual innervation of this box. We've got both sympathetic and parasympathetic controls. If acetylcholine is docking to muscarinic receptors, we're going to have this rest and digest. If epinephrine and norepinephrine are docking to these adrenergic receptors, we're going to have fight or flight. With the risk of making this thing even more crowded, what I'll try and put next to each of these receptors is the fact that when we see these like alpha 1 or beta 1, any of the ones, alpha 1 and beta 1, they tend to be excitatory receptors. So they're going to cause an excitatory response. I typically put a little plus sign next to the ones that are excitatory. And then next to the inhibitory receptors, these are alpha 2 and beta 2. These tend to be inhibitory receptors. So if a chemical signal is triggering these receptors, it's going to elicit an inhibitory response. And if you are in fight or flight, this beta 3 is going to trigger the melting down of adipose tissue. If we're in fight or flight, we want to burn through adipose tissue and use it as a fuel source to generate all the activities that we're doing. This is the case study that we do next time. We're going to learn that alpha 1, these are adrenergic receptors found on most blood vessels. There are a few blood vessels that are instead of being lined with alpha 1, they're lined with beta 2. It's an inhibitory receptor. We see these on the coronary blood vessels. So these are the blood vessels that go to the wall of the heart. And we also see them in blood vessels that go to the skeletal muscles. So like we said, alpha 1, most blood vessels are going to constrict. So the blood vessels that go to the digestive system are going to constrict. Blood vessels that go to the heart wall and skeletal muscles are going to dilate. Alpha 2, 2 is an inhibitory signal. These alpha 2 receptors are going to be located on digestive glands, like the pancreas, for example. If we are in fight or flight, there's going to be a signal sent to the digestive organs to inhibit them. So your digestive signals are going to quieten down. Again, if you're in fight or flight, it's no time to ramp up digestive activity. So that's shut down. And at the same time, digestive activity is inhibited. We can see beta 1 receptors. These are going to be located on the heart wall. And I feel like I'm already jumping into the meat of next lecture. But since we have this slide out, maybe just to the right of this, next to alpha 1, if you can just write most blood vessels have alpha 1 on them. And next to alpha 2, you can just put digestive glands, like the pancreas. These are going to get inhibited during fight or flight. Beta 1 and beta 2 have something to do with the heart. Beta 1, these are going to be receptors that are found in the muscular wall of the heart. So I'm going to try and use this one model to demonstrate the difference here. Beta 1 receptors are in the myocardium of the heart. And what they do is they cause the heart wall to beat more forcefully. So heart contractions are going to increase, and they're also going to be more forceful under sympathetic signals. Again, the heart wall has beta 1 receptors. Sympathetic signals are going to release epinephrine and norepinephrine. When epinephrine and norepinephrine dock to beta 1, it's going to cause the heart to beat more forcefully and more frequently. And if the heart is working harder, it's going to need more blood supply. So that's where beta 2 and beta 1 kind of go hand in hand. As the heart is working harder, beta 2 is allowing more blood flow to the walls of the heart. So all of these coronary blood vessels have beta 2 on them, and they relax and allow the wall of the heart to get hit with even more fresh blood. So beta 1 causes the heart to pump harder. Beta 2 allows more blood flow to the wall of the heart. And then we have beta 3, which we said just activates either the storage of adipose tissue, or in this case, under sympathetic signals, it breaks down adipose tissue. I would put a star next to this slide because we drew it. I'm sure I'll put it on the board next time. It really kind of explains the anatomical difference between our somatic and autonomic division. Remember, in the autonomic division, there's always 2 motor neurons, so we can use the term preganglionic and postganglionic neuron. So if there are 2 motor neurons, remember, which neurotransmitter is released from the preganglionic neuron? It's always going to be acetylcholine. But the neurotransmitter released by the postsynaptic neuron will tell you whether or not you're still in the parasympathetic division or whether you're in now the sympathetic division if you're seeing epinephrine or norepinephrine. Here's some practice ones. Let's look at this. Well, I said practice, and the answer is right there. So let me cover this up, and let's just look at what we're looking at here. We're seeing a preganglionic neuron that has a really long, we've got a long preganglionic neuron and a short postganglionic neuron. That's one clue. And then another clue, maybe the best clue, is that we can see acetylcholine being released at both presynaptic neuron and postsynaptic neuron axon terminals. If postsynaptic neuron is releasing acetylcholine, you know we're still in parasympathetic division. This is rest and digest. So short postganglionic neurons still releasing acetylcholine. This is parasympathetic signals versus what we've drawn here. I could cover this up. And we can see what we've got is a short preganglionic neuron and a relatively longer postganglionic neuron. That's one clue. The other clue, that's the dead giveaway, that our postganglionic neuron is now releasing norepinephrine or epinephrine. Now we have switched over to the sympathetic division, and this different chemical signal is going to cause completely different responses from those same target organs. So if anybody's ever known anybody that's had a heart attack, they've gone into or had heart problems where their heart is specifically having a heart attack, going into the emergency room, it's not uncommon for you to hear people being given beta blockers. Right? Beta blockers, what? Thinking of something there? So beta blockers specifically are blocking beta-1 receptors on the heart. And can, if the heart is beating too forcefully and too quickly, you can hit it with beta blockers and slow the heart down. Here's where we're getting into your stuff. How did the medications affect these receptors? And how is that going to, like if we're trying to affect heart rate, if we want to increase heart rate, then it's adrenergic receptors. If we want to decrease heart rate, we're looking at muscarinic receptors. We've come a long way in one day. I feel like we've got this graph on the board. I didn't think we would get all the way to nicotinic and muscarinic receptors, kind of separating cholinergic and adrenergic receptors. We likely will put this back up on the board next time so that we can navigate the case study questions. Deep breath. We've gotten halfway through Unit 5 information right now. We did a sketch of the 12 pairs of cranial nerves. I'm not going to require that you do that diagram, but there will be questions about knowing which cranial nerve does what. So knowing the names of the 12 cranial nerves would be one thing, and then knowing how to navigate some of the multiple choice or matching questions where you have to match function with cranial nerve. Then the other diagram we did today was this big one that really tried to separate not only somatic from autonomic division, but really the autopilot, the autonomic division, how there are two motor neurons in this autonomic division branch, and there's some anatomical differences between them. Long preganglionic neuron versus short postganglionic neuron in parasympathetic, and then that's opposite. Even though this doesn't really do a good job of showing it, there's a shorter preganglionic neuron and a longer postganglionic neuron in sympathetic division.